We are finishing up Unit 2, Part 2B PowerPoint right now. And this is a quick overview of the picture. I just want to point out again that you should be comfortable knowing that at A, conjugation is occurring, at B, transduction is occurring, and at C, it is transformation. Each one has something unique in the picture to give you a visual guide. For the transformation, the two are being attached by a pili. For transduction, there is a bacteriophage that is helping to deliver the genetic material from the donor to the recipient. And in C, dead implies that this is transformation. And again, please remember the gene is resistance, which is short for an R plasmid, or can be shortened saying an R plasmid. We're going to switch gears a little bit right now and talk about genetic engineering. So a simple definition is this is when there's a very hands-on, deliberate modification for specific characteristics in an organism, and it's done by manipulating genetic material. So the genetic material in a lab is manipulated to purposefully cause some type of change. So this is often done at not only research labs, but at the university level as well. You do, in fact, hire a technician. There's a ton of equipment attached to it, and it is a pretty expensive process. At TGen downtown, as well as at the Mayo Clinic, there are a lot of genetic engineering research projects ongoing at the moment. So we're going to tie together a little bit of how this genetic engineering is done based on things we have already talked about in the past. This is a bacteria cell right here. This is the bacterial chromosome and the plasmid. Together, remember, the two are referred to as a genome. Notice that the plasmid was taken out of the bacteria. And here we have some other type of DNA. This could be either eukaryotic or prokaryotic. And within this DNA, you want a small little portion, in this case, this little green part, essentially cut out. And then that little portion is to be embedded into the plasmid that came from the bacteria. So now this plasmid will be multiplying with this embedded sequence of DNA and showing those characteristics as it's been modified. And this is a simple explanation of genetic engineering. But the question is, how do you actually get this piece of genetic material, whether it's eukaryotic or prokaryotic, out? And once you get it out and into the plasmid, how do you then put it back into a bacterial cell so you can start replicating it and expressing this trait that you have genetically modified? And then how do you, in fact, visualize that this was done correctly? So the way you do this is you use something known as a restriction endonuclease. Remember, when there's an ACE, that implies that it's an enzyme. So a restriction endonuclease is an enzyme that's made by bacteria and has a unique property of being able to cut DNA at a specific sequence. So you can look at a restriction enzyme as a pair of scissors that is cutting bacterial chromosome at a specific area. Now, I want to explain to you something known as sticky ends. This is a phenomenon when you work with genetic engineering and the splicing or cutting of bacterial chromosomes. You have a specific region that you actually want to cut out, but at the same time, you need a little extra on the end. So the example I tend to give students is if you want to sew a pillow and you know you want this pillow to be 1 foot by 1 foot. When you go to the fabric store, you're not going to order 12 inches by 12 inches. You're probably going to order 13 or 14 inches. You want a tiny little bit on the ends, just known as sticky ends, which is exactly what this is. So you have your specific sequence, and then you want a couple of bases at the ends to make sure that you're manipulating and inserting exactly that specific sequence that you want into your receiving bacterial cell. So now once that particular special sequence of DNA has been put into the bacterial chromosome, you then need to visualize, did it actually get in there? You want to know if it's the correct sequence, and you actually need to be able to see the DNA to check this. You're going to want to run something known as a gel electrophoresis to visualize the DNA. And what this is, it's electrophoresis is the movement of the DNA or these charged particles in some type of gel, and then you look at it under a UV light. So first you create the gel. This is similar to how you would make agar, which we've talked about earlier in the class and lab. And then you put something known as a fork into it while it's still wet. And then when it dries, there's all these little openings on the end. These openings are known as wells. And then you inject your material into the wells. You do what you've always been told since you've been a little kid. You don't add electricity and liquid, but nonetheless for electrophoresis, that's exactly what you do. You put the gel into a plastic running apparatus, and then you add a liquid buffer. After you add the buffer, you add an electric current. And this electric current allows the DNA to run down the gel. In science, you're never able to run an experiment without some type of control, because if there's no control, you don't know if the test is correct, and there's no basis of comparison. So the first lane is always something known as a marker or a stepladder. If you look at the bottom picture, you can see that this one is nicely spread out. So you actually purchase this from a company, and DNA is measured in kilobases. So closer to the original well is larger. So essentially we can say this large one is maybe 10,000 kilobases. Somewhere in the middle here, let's say it's about 5,000, and all the way down here, maybe it's about 500. So in a gel, when you are running DNA, the smaller particles run further. The larger ones get stuck up towards the top closer to the well, because the larger the particle, the harder time it has traveling through that gel-like substance. So back to the beginning a little bit. So here, let's look at the third picture. You are loading your stepladder. And in these two, you are loading your experimental DNA that you cut with the restriction enzyme, so you have a basis of comparison. Here you have now added the electric current, and notice the DNA is running through the gel. There is a minus sign up here and a positive sign down here. DNA is negatively charged. If you think of a magnet or opposites attract, and the same charge will repel one another, when you put the DNA into that particular well by the negative side, it will travel towards the positive. In a gel box, if you accidentally reverse it, you know right away, because as soon as you add the electrical current, it starts to bubble. So now you've added the current, and it is starting to separate. On average, you would run a gel 120 to 140 volts for, give or take, an hour, and then it separates. You do need to watch it. There are loading dyes, because DNA and the stepladder at times are clear color. So you would add a gel, in this case it was blue, it could be orange or green. The purpose is so that you can visually see a color as it's running through the gel. If you leave the electric current on too long, if we had a picture of a gel right here, it would look completely clear, because you essentially ran them all off of the end of the gel. So my question for you would be, approximately how large do you think these two DNA fragments are? So remember, with the stepladder, we said up here is about 10,000, here's about 5,000, and here is about 500. So maybe we can say this is about 750 or so kilobases. Notice how the two are the same, so that's a pretty good indication that your sample is accurate. You know how large the sequence is that you wanted to cut using that restriction endonuclease. So if that sequence is around 750, and this is the gel that you're looking at, you know that you've done your work correctly. It's also important to keep in mind that restriction endonucleases, there are many different kinds. There's one called EcoR1 and MBO. You don't need to know any of these. Just please understand that they cut, all restriction endonucleases cut at very specific sequences. You can actually purchase these from companies when you know which sequence you want to take out. When you think of genetic engineering in a farmer's world, maybe they want to genetically engineer crops that have pest resistance built into their genetic material, or something to make the crops grow bigger and faster and stronger. for turning over profit, you'll often hear that things that are genetically modified are healthy or not healthy. If you've heard of cotton candy grapes or have ever eaten them, that is genetically modified. Actually, most foods are, and everything with corn in it is pretty much genetically modified. In the United States, there aren't very strong laws in having to tell the public when something is genetically modified, but in Canada, 1% is the rule through the government that it has to be stated on the labeling that something has in fact been modified. My daughter likes something known as grapples. If you've ever eaten one, I don't know, I think in the fall is when they come in season. Looks like a perfect apple, but when you taste it, it's been genetically modified with grape. So it tastes like a grape, even though it looks like you're eating an apple. So now we've had our DNA sequence. We have cut it with the restriction endonuclease. We have done a gel electrophoresis, which the purpose of a gel is to be able, I'm going to write this down for you, gel electrophoresis. The purpose of that is to be able to visualize DNA. It's a way for you to see if that cut that you made and all of the genetic engineering work is correct. But at this point, you have a very tiny bit of DNA, and that's not enough. So you need to make a lot more, assuming this is for a crop or something along those lines. So this picture is a repeat from where we started. You have the plasmid taken out, some gene of interest that you want placed into the plasmid. You want to then visualize using gel electrophoresis that the cut you made with the restriction enzyme, which is again the scissors essentially to cut the DNA, and then the last step is you want to amplify that DNA or increase the amount that you have. This is done by a process known as PCR. For your exams for PCR, you need to know the steps and be able to explain what's happening. So that's coming up on the next few slides. So PCR is short for polymerase chain reaction. This process is used to amplify or make more of DNA. So again, you have a little bit that you made through genetic engineering, and now you need a larger amount. This machine down here is known as a thermocycler, which completes the different cycles of the PCR steps. So I need for you to be familiar with the 3 steps and understand what's happening at each one. So at letter A, or the first one here, this is the first step, and it's known as the denaturing step. So remember, DNA is double-stranded, so you heat this DNA within the thermocycler to 95 degrees, and it will actually separate the 2 strands. Then the second step at letter B is known as anneal, or annealing. That basically means sticky. Remember from Unit 2, the beginning portion, when we talked about replicating DNA, there was always a primer. So here the temperature is dropped down to 75 degrees. The primers that are specific and complementary base paired to the sequence that you want to replicate, which is that piece that you use the restriction endonuclease to get into the plasmid, it nicely attaches to that. And then the final step at C is called extend. Extend is the growth period. So here the temperature now ramps back up to 72 degrees Celsius, and there's something known as a Taq polymerase, but just remember there's a polymerase that grows and synthesizes the new strands. So you are making many copies of that initial gene of interest that you are trying to amplify or replicate. When you do PCR, notice how it's going from 95 degrees down to 75, and then back up to 72 when it goes from the anneal to the extend portion. Traditionally, when you do PCR, there are 30 cycles of this, which means 30 times it needs to run through step A, B, and C. And in order to do this and make life a little simpler, you load the tubes into this PCR machine and you set it to run the 30 cycles. You set the temperatures and all of the other information, and you essentially hit start, and this will run it for you to complete the cycle. Back in the day, you used to have to put oil or what's called an oil overlay into each of these tiny little tubes. They're about the size of half of your pinky nail, and then manually go from 95 degrees Celsius to 55 to 72, but you still have to do it for all 30 cycles. So you can imagine how difficult that was relative to using a simple thermocycler. I've written the steps out for you here. Let me just write a little bit of information next to each one. So first one, to denature. This is done to separate the strands, or the DNA strands, because again, DNA is double-stranded and you need it to be simple so that you can hit the second step, which is anneal. And here the primers come in, and what they do is they attach to the sequence of interest, or the area of the DNA that you are looking to replicate. And finally, the third step is extend. This is the growth, where all of the DNA at the gene of interest is replicated. Now please realize the amount of liquid that is in the tube that you originally put into the thermocycler is not actually made more. It remains the same amount. It just concentrates with the DNA inside of this amount. So when you work with DNA, you use micropipetters and microliters, drops at a time in very small amounts, but it becomes very amplified and concentrated once you run it through the PCR process. So if you want to pull up an animation of PCR, you can actually do that with a quick Google search. Same with gel electrophoresis, so you can see it happening. But to put this entire thing together, again, you have your bacterial cell right here. You have the plasmid come out. Then there's some type of DNA. This could either be eukaryotic or prokaryotic, but this is essentially your gene of interest, which you then want cut with a restriction endonuclease and spliced into the plasmid. At that point, you're going to run an electrophoresis. The purpose of that is to visualize the DNA, and finally, after you visualize the DNA, this picture above is denoting that you are running a PCR, and the purpose of that is to amplify, or to essentially make more DNA and concentrate it. So now back to genetic engineering. These steps right here are exactly what we've been describing. But the last portion of genetic engineering is we want to discuss a little bit how is this used and where do you find this, or what's known as applications to genetic engineering. These are all examples of how they're used. So we would harvest copies of the gene. For plants and animals, I mentioned earlier that you want to eliminate undesirable phenotypic traits. So let's say you want to involve a gene or specific gene of interest through genetic engineering so that your crop is pest resistant, or you want to create a beneficial type of trait. You want that apple to grow faster and larger and more red. Another reason would be to produce vaccines, antibiotics, hormones, enzymes. So this picture is talking a little bit about pest resistance being inserted via plasmids into plants. Bacteria is also used to clean up toxic wastes. When there are oil spills, they use bacteria because it has the ability to break down oils. Another place that it's used is inside of human growth hormones, as well as something as simple as using enzymes to prepare fabrics for clothing and other manufacturing needs. So please realize that genetic engineering isn't just bacteria or crops. It's for environmental cleanup and pest resistance and all kinds of hormones, and some types of vaccines have genetic engineering involved with them as well. So you want to be able to know the terms from the first part of this second PowerPoint, which are restriction endonuclease is to cut, electrophoresis is to visualize, PCR is to amplify, know the steps of PCR, and everything this page and moving forward are pretty much all of the applications. So how do we actually use them? Please remember the term applications, because you will be seeing this on your exam, where you're matching a couple of examples for genetic engineering as a whole.